Do you know guys, bacteria can literally upgrade themselves by collecting the DNA, the free DNA from the external environment. Does it seem to be like a science fiction? No, actually it is a transformation. It's a process which occurs within the prokaryotes. Let's see the first step. See guys, whenever a bacteria was dead, they are going to release the genetic material into the external environment. Then this DNA, we are going to call it as a naked DNA. This naked DNA is going to be uptaken by some bacterial cells. Those cells we are going to call them as competent cells. But what actually the competent means? See, if any bacterial cell, guys remember this, not all bacterial cells are the competent cells. There are some bacteria like Bacillus and Streptococcus pneumonia, and such type of bacterial cells are going to have, they have the capacity to uptake the free DNA from the external environment. Such kind of bacteria only can be called as a competent cells. See here, this competent cell is going to have a DNA binding receptors. Like not all the bacteria are going to have these receptors on their surface. So due to the presence of these DNA binding receptors on their surface, these receptors are going to uptake this naked DNA from the environment. The third step is the DNA binding, where the naked DNA is going to bind to the DNA receptors which are present on the cell surface of the competent cells. Later, the fourth step is the DNA uptake, where among the two strands of the DNA, one strand is going to be degraded by the enzyme called nucleases. The other strand is going to cross the cell membrane and enter into the cytoplasm of the competent cell. Now the DNA has entered the bacterial cell, but entering into the bacterial cell is not going to be enough, right? Whatever the DNA has entered into the bacterial cell, it should become the part of the bacterial chromosome. Now, it is going to happen by the homologous recombination. But what actually this homologous recombination is? Here guys, the incoming DNA is going to find its similar sequences and it is going to integrate within that in this way. But which enzymes are going to helpful for this recombination? Rec-A protein is going to help. Rec-A protein, this protein is going to help for the recombination of the free DNA, the naked DNA with the bacterial DNA. Guys, if the incoming DNA is a plasmid, for suppose it can be a plasmid too. So, it is not necessary to undergo the recombination procedure because the plasmid can exist independently and replicate independently within the competent cells. Bacteria is undergoing the complete transformation process. But what it is going to gain, guys, these are the some traits where the bacteria is going to acquire. The first is a resistance towards the antibiotics. Second is it is going to have the ability to produce the toxins. Next is it is going to have a new metabolic pathway. The fourth is it is going to increase its survival in the harsh environments. And fifth is increased virulence. In this way, the bacteria, if it is undergoing one proper transformation, it can change its whole behavior. This is a complete process of the transformation which is going to occur within the prokaryotic organism. If you like the video, subscribe to BioSimplified.